Hello, everybody, and welcome to today's fascinating medical sciences lecture titled Making Inroads into the Early Diagnosis of Lung Cancer. Thank you very much for all of you joining us today. My name is Seita Diop. I am a current MSci Medical Innovation Enterprise student here at UCL, and I'll be your chair for today's event. Today's amazing lecture will talk about lung cancer newer treatments and how lung cancer can only be improved by early detection. We want to keep these lectures as interactive as possible, so please make good use of the Q&A function on Zoom throughout to submit your questions for our speaker, Professor Sam Janes. And the hashtag to use on Twitter for this event is hashtag FMS Lectures. That's hashtag FMS Lectures. I am delighted to now introduce our speaker. We have Sam Janes. Sam is the Director of Medicine at University College London, a division within the Faculty of Medical Sciences that encompasses 8 departments, over 20 centres, and around 100 principal investigators across 3 campuses. So a lot there for Sam. His laboratory research, funded by the Medical Research Council and Cancer Research UK programme, grants focuses on the airways and examines how both normal hemostasis and the earliest development of cancer. He works as a respiratory consultant at UCLH with a particular interest in lung cancer, mesothelioma, interventional and diagnostic bronchoscopy, and early lung cancer detection. He has been head of UCL Respiratory, vice chair of the National Clinical Expert Group on Lung Cancer, and the Faculty of Medical Science, vice dean of research at UCL. Thank you so much for joining us today, Sam, and I'll hand over to you for your lecture.
Great. Thank you for the lovely introduction. So I'm going to hopefully persuade you all today that to really crack lung cancer, we need to just focus absolutely everything we can on early detection. And because I'll be really nervous and desperately eager to finish at the end, I thought I'd thank my funders first. So I'm extremely lucky to have had great funding the last 10 or 15 years. And I should thank the Wellcome Trust, who funded me for around 10 years. And then more recently, CR UK and the MRC are funding my current programs. It's kind of traditional now for particularly medics who tend to have quite a few conflicts to do a conflict slide. So I did one. But the one I should point out really is this grant income from GRAIL. So I'm going to talk about a study today called the Summit Study, which I think is an incredible study. And actually, I'm extremely grateful to this American healthcare company called GRAIL, who have funded it, even though it's run by UCL. So just to give you a little bit of background first about lung cancer. Lung cancer, it's a tricky disease, and sadly, it's still the most common cause of cancer death in both men and women. So many of us think of sort of prostate cancer in men or breast cancer in women, but actually lung cancer is by far the biggest killer. Sadly, there's about nearly 100 people a day that's still dying in the UK from lung cancer. And the really tough thing is overall, there's only around 5% of people will survive 10 years following a lung cancer diagnosis. So the statistics are all pretty gloomy. And this is quite a complex slide, but we'll talk through it because it's really important. So lung cancer, obviously, it's within our lungs. And when we diagnose it early, we call it stage 1 lung cancer. So a stage 1 cancer is where you just have the cancer in the lung. And because it's stage 1 and it's just in the lung, that means we can actually give really good treatments. So you can have surgery or you could have radical radiotherapy. We would really hope to cure you if you have stage 1 cancer. Stage 2 cancer is slightly tougher to treat, but still curable. Stage 3, that's getting quite tricky. And stage 3 cancer is where perhaps it's spread to lymph nodes or lymph glands in the middle of the chest, a bit like a breast cancer might spread to the glands in an armpit. And then finally, stage 4 cancer is where it has spread elsewhere in the body. And if you look at this graph, what you can see is, firstly, if we look at just these bars, this is the incidence of the different stages. So you can see, sadly, stage 4 is by far is probably more than half of all lung cancers that I see in my clinic and we see in the UK are stage 4. So the cancer is spread. And I think generally we would say that that's likely to be an incurable cancer. So that's really tough. And then what these lines look at are our survivals. So we can see if we look perhaps the middle line, this is a 5-year survival. So the 5-year survival for stage 1 is okay. So the percentages are over on this side of the chart. But you can see when you get down to stage 4, the 5-year survivals are terrible. So what we need to do is to be diagnosing lung cancer, as with other cancers, early. We need to diagnose it when it's stage 1 and most likely to be cured. So are we making progress in cancer generally? Well, actually, again, this slide's a bit sobering. So I'm going to start a little bit sobering, but hopefully leave you full of hope by the end of the session. So this is the 5-year survivals and what's happened over different decades. So we have 2000 to 2004, 2005, 2009, and then 2010 to 2014. And you can see there are little improvements, but this is our percentage survival. So in the UK is sadly worst out of all these countries. We've chosen these countries because actually they have the best data. So these countries have great data, which allows us to look at overall survival. But you can see we're struggling to keep up with perhaps some of our similar countries. So early detection and treatment sounds obvious. But actually, there have been some great advances in medical treatments recently that we use for stage 4 cancers. So we're using new targeted therapies and we're using new checkpoint inhibitors, immune checkpoint inhibitors, which are undoubtedly making improvements. But there are 2 problems with these. First is the cost of treating lung cancer, as with some other cancers actually, is becoming unaffordable. So here we have American data. So undoubtedly, the costs are much higher. American healthcare costs a lot more than UK healthcare, but the drugs will be the same, similar or same prices. And what you can see here is in 1970, and this is the cost per month of treating lung cancer patients, the average cost was about $100. Whereas in 2017, you can see we're now breaking through $10,000 a month. So that's huge prices. And so this is like a logarithmic scale of cost. And actually, I think it might well keep continuing because in the future, we hope to have cellular therapies or combined therapies where costs are just going to keep going up and up and up. And sadly, I think the UK and a number of other health systems just simply will not be able to afford these treatments. The second point is actually, I believe if you invest in early disease, you really make a difference. But actually, when you invest in late disease, it's really hard to make any improvements. So again, this is a bit of a complex graph, but what we have are the survivals. And again, we have the stage of the lung cancers at the bottom. So when we look first at stage 1, we can see in 2004, which are the blue squares, the survival for stage 1 disease was about 72%. And then over a decade, it went up to around 84%. And what did that actually was a very simple intervention, and that was an audit. And basically, every hospital in England had to record its lung cancer surgery for stage 1 disease, and any hospital with very low rates of surgery or treatment basically got a letter saying, you're outlying a little bit, perhaps you should be treating or operating on more cancers. And what happened, targeting and treating early disease caused an improvement. Now, if you look at the stage 4 disease, over a decade of new drugs and new investment, you can see actually overall for all those patients with stage 4 disease, we made no impact. Even though some individuals will have had great benefit, perhaps many months of benefit from different treatments, overall it makes very little difference. So what will happen perhaps in the next decade? Well, I believe it will be the same, even with new therapies such as checkpoint inhibitors. And actually the 2018 data, it's moved up a little bit, perhaps from 16 to 17%. So I've sort of said cheekily here, the blue circle is where I think we can make massive improvements. However, the red circle is where we put all our investment. So this is where all the NHS money goes on drugs, and this is where all the research money goes on developing therapies for metastatic disease. And yet it is nearly impossible to make a difference. So I'm interested in early disease, and I've got two bits to my talk. So the first bit is going to give you a little bit of actually very patient-based, but a little bit of science, which I'll go through slowly. So I hope you get a little bit of a few exciting things to think about. And then the second bit is going to be about CT screening. So first up, what we have at UCLH is probably the world's largest cohort of people with precancerous airway lesions. So this is a picture from a telescope that goes down the throat when a patient's asleep, and we can look around the lungs. And when we switch on a blue light, we see these little purple patches, which are precancerous lesions. So I've got an example here. So basically, this is us doing a telescope test in a patient going down the windpipe. So at the bottom here, you can see some tracheal rings. So that's the hard bit of our trachea at the front. And then at the back, the trachea is floppy. And then here we have our two lungs. So we're going to go down. We're actually going down into the left lung. And what I want you to see really is that everything looks pink and healthy. And we've just bumped into the airway there. So we caused a little bit of bleeding there. But now we're going to switch on the blue light. And what happens is abnormal tissues fluoresce differently. So I hope what you can see here quite easily is this angry purple area. And that's a precancerous lesion that we call carcinoma in situ. So it's like they're cancer cells, but they're stuck in situ. They're not behaving like a cancer. They're not spreading. And you can see us going in and taking a biopsy of that lesion. And what we can do with that biopsy is then take it back to the laboratory to have a look at it. So firstly, our pathology team say that, yes, this is a carcinoma in situ lesion. But secondly, we take it back to the lab where we can start working and investigating it. And I think one of the really interesting questions that we've always had is when we see these carcinoma in situ lesions, we don't understand why around half of them progress over time, whilst about a third or maybe a bit more regress. So actually these carcinoma in situ lesions disappear. So I'll say a huge thank you first to these 4 students and postdoctoral fellows working in the lab who did much of the work. So Chris, Vitor, Adam, and Henry that I'm going to show you. So basically I show you here 2 patients. So patient 1, we're doing one of these bronchoscopies, these fluorescent bronchoscopies every 6 months. And then eventually what our pathologist says is, Sam, this patient, now this isn't carcinoma in situ anymore. This has progressed to cancer. So they need treatment. Whereas our second patient here is one of our regressive patients. So we're taking our biopsies, seeing carcinoma in situ, and then we see that it's actually regressed. But we keep biopsying to check that it hasn't come back. So we've called these little biopsies just before they progress or just before they regress, we're calling them the index biopsies. And these are the ones that we've analyzed to try and work out why these ones progress and why these ones have regressed. Are there some molecules or genes in these lesions which are predicting that whether you progress or regress? Because if we understand that, we might be able to develop novel treatments. So the first little bit of data was actually, we were just interested in our carcinoma in situ lesions overall. And what sits here are 10 genes that are most commonly mutated in lung cancer. And this is lung squamous cancer. We've called it LUSC for short. But what you can see is actually the exact same genes are mutated in the carcinoma in situ. So, exactly the same numbers. And we were really surprised. We assumed the cancers would be much more mutated. So our second question was, is there a difference between the progressive carcinoma in situ and the regressive? And actually there is. So the regressive ones do have less of these mutations, but interestingly, they still do have driver mutations, and driver mutations are the mutations in the particular genes that we think cause cancer. So, we've got one carcinoma in situ lesion here, has 5 driver mutations, and yet still is able to regress. So we then looked at what we call the transcriptomic and epigenetic, and the details of this, again, don't matter too much. But what I think you'll be able to see is, firstly, when we looked at the protein, the sort of RNA in these patient samples, we can see the regressives and the progressives look completely different. So these are all, every little horizontal dash is a different protein, and basically you can see these enormous differences. And then actually the epigenetic signatures were also different. So again, we have our progressives compared to our regressives and normals. And you can see the clustering in these genes, meaning that there are definitely signatures within these lesions that will determine whether they progress or regress. And there's going to be particular genes and pathways. Now, if you have DNA mutations in your cells, what should happen is your DNA is abnormal. So you might, not all the time, but you should, probably a lot of the time, you will produce an abnormal protein in that cell. So let's say it's on your skin, or actually let's stick to lungs. So you smoke and you created DNA damage in your cells. This should lead to an abnormal protein. An abnormal protein should be presented on the surface of the cell, and that means that immune cells that are passing by should see those abnormal proteins and think that's not me, that's not right, and they should kill the cell. So actually, as humans, we shouldn't really generate cancers if our immune system is working exactly as we want it to. So the next question we had was, okay, are there problems with the immune system in these pre-cancerous lesions that predict whether these lesions will progress to cancer? So basically, your lesion, the cells start adapting desperately to try and help themselves survive, enabling them to eventually progress to cancer. And this is extremely detailed, but what you have here are the 62 genes that are involved in the presentation of abnormal proteins to the immune system. And each one of these colors or boxes or dashes is an abnormality. So you can see actually these lesions are doing absolutely everything they can to avoid the immune system detecting them. So what do we believe is happening for lung cancer to form? Well, first, there is indeed a buildup of genetic damage, pretty much that matches cancers. But secondly, I think you have to have this rapid adaptation to try and make the immune detection fail. So basically, this then means that you can progress to form a cancer. So I suppose the next question is, how early does this start? I've shown all these mutations in the carcinoma in situ lesions. What about normal tissue? And this was amazing work done by Kate Gowers in my lab, working with the Sanger Institute and Peter Campbell's team. And what Kate did was she took biopsies from patients. She then digested them into single cells and grew up these single cells as little clones. And when we could get a little clone, that would mean we could get enough DNA to whole genome sequence the clone. So in theory, we're now sequencing single cells from people's airways. Now, this is, again, incredible data that Kate has put together. But basically, she found that, if you take cells from a child, they have very, very few mutations, just as we would expect. So you can see one cell here seems to have quite a few mutations. If you look at never smokers, they do actually have quite a few mutations, probably nearly 1,000. So it works out actually every cell in our lungs is getting about 22 mutations per year, even without smoking. So that's somewhat sobering. But then look at the smokers, ex-smokers and current smokers, and you can see lots and lots of some of these cells have like 15,000 mutations. This is as much as a cancer. So somehow these cells are surviving in our airway and not forming a cancer. What's quite interesting is the number of mutations is really variable. So some have, 10 or 15,000 mutations, whereas others have pretty well the normal level. So this suggests that actually some cells are either resistant to smoke, or they're hidden away somewhere and not being affected by smoke. And these cells, they can't form cancer because they don't have the mutations. But perhaps most interesting is this. So this, again, is a little bit complex, but we'll go nice and slowly. So here we have the fraction of cells with near normal mutational burden. So what these are those cells at the bottom with no mutations, or just the number of mutations you should get with your age. So if we look at the smokers, this is about, I don't know, 3 or 5% of the cells have almost no smoking related mutations. But look at the ex-smokers. And just to really sort of exaggerate my point, we'll look at this 71-year-old, the third bar in. Over half of this heavy ex-smokers' cells in their airways have no sign of tobacco damage. So I think that's really cool. And that's probably the best evidence that any smokers out there should really try and stop, because it will make a difference. Because basically what seems to happen is your airways repair using these cells with almost no damage. So I wonder whether we could see a future where we could manipulate our lung and sort of expand these low mutant cells, thereby reducing cancer incidence. So I'm going to finish just with, I think, a really super important story. And this is something that you will all see happening around you over the next few years. So this is low-dose CT screening for lung cancer. So this is a CT scanner. So many of you will have seen CT scanners. Some of you might have had them. And basically you lie on this and sort of go into this donut ring, and it gives you lovely views, which are like being salami sliced. So it's like salami slicing the lungs down. You get incredible views and incredible details of the lung. And what a very large study in America, nearly 54,000 people, in a study called NLST, it was shown that lung cancer screening works. It saves lung cancer lives, and actually it reduces overall mortality. So this is a really, really effective way of screening for lung cancer. Now, I was really keen to screen in London. I needed a trial because the NHS wasn't going to pay for CT screening. And one of the things I was interested in is the challenges to recruitment, because the effectiveness of screening is only, it's only effective if those at most risk turn up to screening. And of course, lung cancer has lots of inequalities, both socioeconomic and smoking related inequalities. So in 2013, there was a study in the UK, so a 2,000-person study called UKLS in Cambridge and Liverpool. But there were rumors that they were really struggling to recruit to that trial. And similarly, in the US, where they'd approved lung cancer screening, only 4% of people that qualified were getting lung cancer screening. So this was creating huge skepticism amongst health workers in the UK, politicians, and really disbelief that CT screening was going to be effective. Because if you only get 10% of people turning up, then it's not going to be a cost-effective thing to introduce. There was also huge fear of what we call false positives, huge fear of incidental findings, other diseases turning up, huge fears about workforce and capacity, just basically huge fears about CT screening, and it was viewed very negatively. So at that point, I had the remarkable good fortune in that I met Jane Wardle. And Jane was a psychologist, and she published 700 or 800 papers on breast screening and bowel screening, and was really interested in really trying to improve the screening process and making sure people turned up for screening. And she was really interested in lung cancer because she was interested that probably lung cancer had to target who we are sort of termed the hard to reach. So they're people often from the lowest socioeconomic quintiles, traditionally sort of rejected by medicine because they often are smokers or ex-smokers. And then we managed to, by huge fortune, recruit these two amazing PhD students. So Sammy Quaife, who now runs her own group at Queen Mary's Hospital, And Mumta Ruparel was a clinical fellow working me, who now has developed her own group, in fact, in Singapore. And what we came up with was this idea of an MOT for your lungs, because we thought actually heavy smokers would understand this as a bit of a checkup, bit of a light touch checkup, just to make sure they're safe for the next year, as they have for their cars. And we also dubbed this the lung health check. And what Sami Quaife built was invitational materials, which was our trial, actually. So we trialed this lung MOT invitational material. And this was relatively easy to read, very low on numbers, which we thought would, and we tested it out and felt it would suit the population that we were targeting. And this was compared against a control material, which was much more like a traditional screening material used for breast cancer or bowel cancer. So what happened? Well, actually, the main point of this trial was that it was a resounding success in that our recruitment rate was an incredible 53%. So 53% of those invited came up for their lung health check and CT screen. So this really sent shockwaves through the lung screening, interest groups and politicians, who then suddenly were actually faced with, actually, this screening not only works, but people want to have it done. Did the lung MOT invitational material actually work? Well, actually, it didn't. There were no differences between the control group and the interventional group, even though there were reports of people preferring the lung MOT. So where were we with lung screening? Well, in 2016, the European trial, which many of the European governments were waiting for, was still some years off. It was supposed to be announced in 2016, 2017. I was desperate to do CT screening in London, and I looked for partnership. And actually, UCLH were also really keen. So they were really trying to see whether they could afford to do some lung cancer screening. But then I had the incredible fortune to meet some investigators from this company called Grail. So they're situated in California. And basically, they have a blood test which detects what we call circulating tumor DNA. So basically, you have a little cancer or any sort of cancer. And actually, cancers all the time are both growing and dying. Some of the cells are dying, and when they die, they pop into the bloodstream and they burst and release their DNA. So, and this DNA, of course, is abnormal because it's from a cancer, so it has genetic mutations. And what they're detecting in this test is the difference between cancer DNA and the normal DNA that is also floating around in our blood. So we designed the Summit study. And so Summit was funded by Grail. It's sponsored by UCL. So it's an academic study. And it was delivered by University College London, the trials unit, and our hospital partners. So 11 hospitals around North Central and East London took part. We recruited 13,000 people, and we tested mainly the feasibility of implementing low-dose CT in a major city, and secondly, the performance of the Grail blood test. And we recruited those 55 to 77, and you have to meet one of two risk scores, which basically are largely influenced by the number of cigarettes you smoked in the past. So Grail built with us 4 CT screening centers. So they supplied the scanners, they paid for the staff, the reading, the computer system. And it was really just a huge task. And this was, fantastically delivered by people employed by UCLH, but working across North Central and East London. And this is our dashboard for recruitment. So I took this screenshot actually just before we finished. So we finished at 13,000 participants. But what you can see here, these little orange bars, are the number of people recruited per day. So it's around 70 to 80 people per day being recruited to Summit until we hit a pandemic. So you can imagine running a massive respiratory screening trial during a pandemic was pretty tough. So we had a lockdown, and then I think this is really interesting. When we reopened, we found it incredibly difficult to recruit. So, you know, people just did not want to leave their homes to come up to a hospital for a scan. We had our second lockdown. Recruitment improved a little bit, but then we closed in May 2021. Summit has been an amazing journey for me and the wider team, and we've done lots of things which I hope will be an incredible legacy for UCLH and UCL, and hopefully collaborators and investigators across the UK. Because not only have the company Grail taken a blood test to check for their cancer test, but we have also collected blood samples from our participants. We've collected all their imaging, and we've collected primary and secondary care data as well. And in fact, CR UK have also now funded the collection of anybody of cancers from anybody with cancer. And so we're already now starting to build on this. So we have Summit, our 13,000 participants, and now we're rolling it into a lung health check program, which is funded by the NHS, and continuing to collect samples from the participants within that. And we're already building in new studies. So we've got some EU and some CR UK funding. And then we hope to start working with other partners, looking at cohorts of people within Summit. So we have a big cohort of people, for example, with COPD, a chronic obstructive pulmonary disease that haven't been diagnosed. So we can see it from Summit because we can see their scans, and we do blowing tests and collect data. So I think this is an incredible opportunity to hopefully deliver early treatments to people with different diseases. So finally, where are we with CT screening? Well, the Nelson trial finally published in 2020, again showing this fantastic mortality reduction. This is lung cancer specific mortality reduction. So at this stage, most European governments have now said that they will fund CT screening. And actually, I think UK and Croatia really lead the way in this. And our National Screening Committee have said that we should be doing lung cancer screening. And actually, the government are indeed going to fund it. So we've already run phase 1, 2, and 3 pilots. And the hope is the original slides were 23, 24. My suspicion it will be 27, 28. We will actually be rolling CT screening out across the whole of England and hopefully the UK. So finally, acknowledgements. I acknowledge the scientists along the way. This is the lung screen uptake team. So again, Jane, Sammy, and Mumta, but it's a huge team that's delivered this. And Summit is an enormous team effort. And I thank everybody who has worked in Summit over the past 4 years delivering it, and actually the 2 or 3 years beforehand trying to design it. So I'm going to stop there, and thank you very much.
Thank you so much, Sam. That was such a engaging and fascinating lecture from you. And also congratulations to you and, like you said, everybody involved in getting this together. I can imagine it was an incredible amount of work, but it's really amazing to see how much you've contributed to just the data in general within this space, but also such an amazing accomplishment to get the NHS to roll this out to a national kind of movement. I think, honestly, congratulations on that. That's a really amazing achievement. I'm going to move on to the Q&A. We have a few questions from our audience. Someone has asked, so you mentioned during the bronchoscopy that bleeding can be caused, and someone was wondering whether the causing of this bleeding, does that have any serious damage to the lungs or to the person's wider health?
Yeah, no is the short answer. So you saw when we took the little biopsy, you sometimes do get a little bit of blood there, but actually it's as though you get a little scratch on your skin. If it bleeds, it's only for a few, 10 or 15 seconds before it stops bleeding. So the biopsying of these lesions is very safe. If you need sort of more involved treatments for a cancer or something, then that can be more difficult, but obviously more important to do as well. So
Absolutely. So you mentioned that it was low dose, or low density CTs that we used.
Yeah.
Someone's asked here, does normal CT work? And if not, why not?
Yeah, so CT scanning is computerized tomography. So you saw the sort of scanner, and the advantage is the disadvantage of CT scanners is that you do get a dose of radiation when you have the scan, whereas an MRI, for example, there's no radiation.
Mhm.
Now, the problem with MRIs is they don't work if there's air. So for the lungs, they're no good. So brilliant at the brain, liver, other things, but no good for lungs. So we're left with CT scans. So then basically the people that make CT scanners, the real drive is to reduce the radiation dose. So there's been huge investment, and now actually we have ultra low dose CT scanners. We also make them low dose because we don't use contrast at all. So again, the use of contrast, which shows blood vessels, increases the amount of radiation. So there's lots of sort of lay ways of describing the amount of radiation. I use one that it's thought, so a low dose CT scan's thought to be roughly equivalent to a transatlantic flight. The sort of dose of radiation that you can get. So most people will feel fairly reassured by that.
There you go. Hopefully that answers your question. So you were showing that chart that kind of showed the different levels of mutation depending on the smoking status of different patients. And we noticed that there was kind of, it seemed at least that there was higher potentially numbers of mutations for those who were ex-smokers versus those who were current smokers. Do you think there's a reason behind that? And someone's asked, so is quitting smoking bad? I assume there is a correct answer to that question, but if you could elaborate a little bit more on that for us.
Yeah, so I think overall there was no, so we only had 9 people overall who were 6 ex-smokers and 3 smokers. Overall, there wasn't a difference. There were a few sort of outlying cells. Remember, these are just, each dot was a cell. So it's, overall, there wasn't too much difference. But overall, actually, the ex-smokers they'd been heavy ex-smokers and often only quit a few years ago. So they may have smoked for 40 or 50 years in some cases. I still think the most interesting thing is the number of cells in your airways, so your trachea and bronchi, that actually end up with no sign of smoking damage at all. So, you know, in that one sort of 71-year-old that I sort of pointed out, over half of their cells had never seen smoke. It's like weird, you know, where have these cells come from? And what we think is that when in our windpipes, we have little glands, and we think sort of stem cells or the cells that produce new cells might be in these glands, and they're a bit protected from smoke. And actually, if you can stop smoking, actually they really happily grow out and start repopulating your windpipes with cells that have not been damaged by smoke. Now, the great thing is, if you haven't been damaged, if you haven't got any DNA damage, then you're not going to form a cancer. So, you know, in that 71-year-old, half of their windpipe now is protected against cancer. And if we could really understand that, you know, maybe there are drugs or inhalers that we could use to really help those normal cells grow out. So then if people came to me as a doctor having smoked for 20 years, I could say, oh, look, you know, take this drug and we can get rid of those cells that are more likely to form cancer. So that's my sort of, you know, next 5-year plan.
I mean, that's amazing. It's amazing I'm going to be watching this space to see the next avenues within that space. That's quite an exciting kind of potential. So another question here we've got is, do you think there might be differences in cell mutations from people exposed to tobacco versus those exposed to biomass combustion? I'm assuming this is someone who lives in the city like me and is worried about things like that. And any of the potential risk factors. Are you stratifying the patients in regards to this risk, or is it specifically smoking that you're looking at?
Yeah, so really good point. So the mutations are different. So there are certain sort of signatures, we call them, of mutations from smoke or different chemicals, and pollution is different. There's been quite interesting work just recently from UCL, from Charlie Swanton's lab, which has looked at lung cancers in never smokers, because we've never really understood why they should get lung cancers. And that, he's got some quite nice data which does suggest that pollution, it might not cause the mutations, but actually the environment just becomes a bit inflammatory for the cells, so they start growing quicker than they probably should do. And then if you're unlucky enough to have a particular mutation, that combined with the pollution in the environment might mean you're unlucky enough to form a cancer. So I think the question is quite correct. They do look like they're different, and that will be much more work trying to understand that.
Okay. Let's see. Okay, so we have a question here asking about sort of multidisciplinary work that goes behind something like Summit and the work that you've been doing. And they've asked, do you think further study into epigenetics will help early diagnosis? And what other kind of interdisciplinary and wider disciplines do you think could contribute to furthering this?
Yeah, so the epigenetics, I think, is really important. So we have that first data on our precancerous lesions, looking at the epigenetics. So epigenetics is basically you have your DNA code, but then the DNA also has other little chemicals on it, that we term methylation. So it's sort of either methylated or it's not, which means genes switch on or not. So the epigenetics is really whether you're expressing the genes rather than whether they're mutated or not. And we do see a difference. So we see a difference in our precancerous epigenetics. And actually the GRAIL test, so the circulating free tumor DNA test, is actually an epigenetic test as well. So yeah, I think the question's right on the money. I think epigenetics is really important. I think one of going back to Summit and the sort of multidisciplinary nature of the team, I mean, it's been fantastic. I've learned so much working. So we work with our sort of statisticians, the behavioral scientists, the nursing team, the trial practitioners. We've got an amazing reception staff. We've got had a huge telephone staff, actually. For the first time in my career, I worked with maybe 10 telephone staff answering calls and doing phone screener checks and things. It's been really wonderful. And I think hopefully the research we're producing is really going to demonstrate that as well. So we've published papers on the psychology of CT screening. We'll do papers on smoking cessation, as well as the traditional radiology or cancer outcomes that you might expect. So it's been a real honor working with such an amazing group of people.
Okay. So a few questions. I'm kind of looking ahead on what you're currently working on. Someone has asked if you are potentially looking if there's a scope to expand the population that are going to be screened with LDCT. This one is specific to the person asking the question. They're 86 years old and they've been smoking since they were 15 and wanted to know whether, like I said, if there's scope to expand the population that would be screened.
Yeah, really, really interesting question. So when you work with screening, so if I screened, obviously, if you screened everybody, I would catch all the cancers. But then you have to work out a few other things. So one, obviously, that costs a lot of money and it's very hard to deliver. Secondly, something like CT screening, there is a harm. So there is the low dose of radiation, but actually there are other harms. So we might detect, for example, we might detect a very, very slow growing lung cancer, and actually that person might end up having an operation that, in fact, if we've never detected it, it may never have affected them, and perhaps they would have died of a heart attack 5 years later. So that's actually a harm then, because you've done an operation that wasn't required. And then you also have to look at your population, so the age of your population, you have the harms of what you're doing start increasing compared to the benefits. So this sounds terribly tough, but if I were to operate on someone at the lowest age, the 55-year-old, they have lower risks from, say, lung cancer surgery. So their risks of undergoing an operation are much lower than, say, a 90-year-old. And secondly, you obviously hope to gain them many years of life, whereas the life expectancy of a 90-year-old might be another 3 or 4 years. So you have to weigh all of these things up. And basically, that's what the National Lung Screening Committee do. So some of the trials, so Summit, for example, we went to, you had to be 77 at entry to the study, so you would then be screened till you were 80. Mm-hmm. I think the national pilots are only until 75. And some of that will be driven by statistics and finance, as well as actually, the harms that might occur to the older population if you screen them and start operating or acting on cancers. And we call that an overdiagnosis. So basically you're overdiagnosing and overtreating and actually harming people. So it's very complex, a complex area.
Yeah, I can absolutely imagine, like you said, it's a fine balancing act between a lot of different elements to figure out exactly who the target population should be. Okay, so we've got a question here asking, are you thinking of joining forces with trials in other countries? And there was another question that linked to that. Are you planning to conduct any studies on, oh no, just that one. Are you planning on joining forces with any other countries or any wider studies?
Yeah, so what we hope, Summit is an incredible, so Summit is the largest CT screening trial in Europe. And one of the real pleasures of being an academic actually is you meet people around the world with the same interest, and they're often incredible people. And I'm very lucky I met Harry de Koning, who ran the Nelson study, which was the European study that we were waiting for and came out in 2020. And so Harry has a really wonderful new program of CT screening, and Summit will now contribute hopefully hugely to that program. And then there's also a real interest in biomarkers, other biomarkers, so blood tests or breath tests or some other test to try and detect cancers early, as well as our GRAIL test. So with our lung health check program, which will be rolling forward now, we're joining an EU program called Prevalung, which is run from Paris, actually. And so we'll be contributing to that. So yeah, I really hope that UCL, UCLH and our North Central in East London will continue sort of really being at the forefront of things across Europe moving forward.
That sounds very exciting. You'll have to brush up a little bit on your French as you go in to venture with the Parisians. Okay, so we have a bit more of a technical question based on some of the things that you've mentioned already. They've asked, could you elaborate what immune checkpoint inhibitors are and any specific details on how they're beneficial in stage 4 treatment?
Yeah, so they were a Nobel Prize winning discovery a few years ago. And basically what scientists found, so it goes back to my point that when you have a damaged DNA, that should mean that when that gene is being read and a protein is being made, the damage will result in an amino acid in the protein being wrong. So when your cell is finished with that bit of protein, all these proteins float to the surface and are basically expressed on the surface of the cell. And they're sort of shown with this sort of antigen presenting machinery. And what should happen is your T cells come along and they check all the proteins. So T cells are really made to find viruses. So if they see a viral protein, they'll go, oh, hang on a minute, this is bad, and they'll kill the cell.
Mm-hmm.
But what should happen if your amino acid is wrong is they should detect that and kill the cell. But what happens, and I demonstrated that, showing that with those 62 genes, you get all these different abnormalities occur. The cells are doing everything they can not to die. And one of the things they do is they upregulate a protein, or probably a number of proteins, called PDL1. And what that protein does is it sort of sticks out of the cell. And when a T cell comes along and sits down, it tells the T cell, do not kill me. It's very clever. So that's what cancers are doing. They're putting little proteins on their surface saying, do not kill me. It's like a sort of cloak, a shield almost. So what these incredible drugs, these immune checkpoint inhibitors do, are they're little antibodies that you inject, and they come along and they recognize these little proteins like PDL1 sticking out of the cancer cell, and they sit on them and stop them working. So when the T cell comes along, it's no longer switched off and it can kill the cancer cell. So they've really changed the face of melanoma. That was the first cancer that they were successful in. It completely changed melanoma. And actually lung cancer is probably the second most successful cancer. So what will happen is, if you are unfortunate enough to have you or a relative or friend have a lung cancer, now in the clinic, every hospital, certainly in the UK, will check for these proteins. And if you have these proteins, then you'll get these checkpoint inhibitors. And you might get just the checkpoint inhibitor, or your doctor might give it with some chemotherapy as well. And what we find is that, sadly, cancer drugs don't work in everyone, and neither do immune checkpoint inhibitors. But what we do find is they're pretty effective. And in some people, they're really effective. So, you can be lucky. And, now maybe for the first time, we're even talking about, I'm hesitant to say it, but even talking about cures in metastatic lung cancer.
Right.
So, they really are game-changing drugs. Game-changing for a few, help a lot. And sadly, in some people, as ever, they make no difference.
So is this kind of one of those places where that we'll see more of an intersection between kind of personalized medicine and the use of like epigenetic and genomic screening to really identify who would be like ideal candidates for some of these drugs that you're mentioning?
Yeah, so I think we would say that, immune checkpoint inhibitors are personalized in that we are looking at the PDL1 expression on the cancer. So if you have no PDL1 there, then actually the chances are you're not going to do terribly well on them. So in fact, I don't, NICE, for example, I don't think fund that treatment for cancers with no PDL1. So yeah, absolutely, another sort of part of our armory that is personalized against lung cancers.
Okay. We have time for a couple more questions. This one's actually from me, not from the Q&A, but I'm curious nonetheless. What do you think are the kind of possibilities for spreading something like Summit and just general more kind of early detection and screening beyond Europe or the developed world? I know I'm from Senegal, and I know in Senegal, I think we have like 2 CT machines in the whole country. So obviously not very many as it stands. What do you think the kind of collaboration is needed, or the future could be for really spreading this to be more accessible wider around the world?
Yeah, I think that flags a few points actually. So there is undoubtedly a problem where third world countries aren't going to be able to deliver CT screening because of its accessibility of CT scanners, workforce costs. So, we as a sort of global physicians need to keep thinking and working on that. And I think that's one of the reasons why we're actually really interested in biomarkers as well. Perhaps blood tests or breath tests might be more accessible. There's actually also a problem, and it's kind of come up already, the age group that it will be offered to in the UK is 55 to 75, I believe. So, we had somebody 84 saying, why can't I be screened?
Mm-hmm.
And that's completely valid, So, and then the other thing with CT screening is it's only those at the very highest risk. So you have to have smoked maybe 20 to 25, a pack a day for 20 or 25 years,
Mm-hmm.
probably is the ballpark region to qualify. Now, even if you ran the perfect CT screening in the UK, where everybody that qualified got a scan, and you've seen in my study, maybe half or a bit less will get a scan. Even if you ran the perfect CT screening program, 60% of lung cancers are actually in people that won't qualify.
Yeah.
So they're in the lower risk or never smokers. So, we've got all these people as well to think about and be developing early detection methods that perhaps aren't CT screening. So that's, a huge challenge, but a challenge that people are really interested in now. CR UK, for example, who fund me, they've created a whole early detection panel over the last 5 years and are really focusing their funding into early detection, because now they too really believe actually that's where we can make the most difference.
Absolutely. Like I said, exciting to see what comes next. I think we have time for one more. Someone's asked, are you planning to conduct any studies on passive smokers and the state of long-term effects on their lungs?
Yeah, really good question. So we haven't. So I think so if you passive smoke, you almost certainly won't qualify for CT screening because it will be felt your risk is too low. So again, I think that's an area where many of us are interested in perhaps other different types of testing. So, we've worked with a company that does breath tests. So you blow into a machine and then the machine tries to detect cancers. And that's the type of test that, if you could get it cheap enough and it was effective, you could then do that in Boots or GP surgeries. And that would really bring early detection of lung cancer for everyone, rather than just the heaviest smokers. And I think, we've got to get CT screening delivered in the UK, work on it globally, and then we've also now got to be really trying to think of new tests where we're actually, all those people that don't qualify, we can be thinking of something for them as well.
All right, final question, which I think is actually a really interesting and timely question, given the prevalence of it nowadays. What knowledge do you have on the effect of different cigarettes or different forms of smoking, specifically vaping, given that it's still quite novel, and that as obviously our knowledge of cigarette smoking and its effects improve, then something new comes along and we have to kind of catch up. So kind of what is that looking like? What's your kind of knowledge around that environment, and anything that you can share with us on that?
Yeah, so I think people's feelings about vaping and e-cigarettes are quite sort of polarized in some ways. So when it came about, I think many people like me who are lung cancer doctors thought, great, this is great, because actually what we don't want our patients to do is smoke cigarettes, because that has 200 carcinogens. We know it gives you emphysema, it's terrible stuff. So e-cigarettes that are just really delivering nicotine and some other chemicals that we're not quite sure what they are, probably safer. It's a good way of delivering nicotine. Of course, what's happened has been predicted by many doctors, is that tobacco companies have brought up most of the e-cigarette companies. We don't know how much nicotine is in them, so they tend to be very addictive. And worryingly, they appear to be targeted towards young people.
Yeah.
And what we're finding now is, if you look at vaping e-cigarette rates in sort of teenagers, they're terribly high. The issue is that these things are full of nicotine, so they will become nicotine addicted. And we know nicotine is as hard to give up as heroin. So that's the rates of quitting smoking are very, very low. So you're probably going to have a lifelong nicotine habit, which is very sad. And then, of course, we don't really know whether that will be a gateway into cigarette smoking or other things. So I think, yeah, it's really worrying. Does it cause lung cancer as much as smoking? No, I don't believe so. Does it cause emphysema as much as smoking? Probably not. Does it cause other things we don't know about? Maybe. Certainly in America, they have a few more problems, but they probably use things a bit more experimentally over there. So yeah, so I don't think the outlook is that great for young people.
Yeah.
Still, in my clinic with my heavy smokers with lung cancer, I say, yeah, switch to e-cigarettes.
Yeah.
And that's kind of what they were for originally.
Understandably. Right, so that is it for the Q&A, and we are sadly out of time for today's session, so we'll have to leave it there. If I could ask our audience, please, if you could give us your feedback about today's session by filling out our survey, which will be sent out to you following today's event, that would be greatly appreciated. And just to finish off by saying thank you so much, Sam. That was such a fascinating and interesting lecture, and I'm sure our audience feels the same. So amazing job, and thank you for all your questions from the audience, and for you, as always, answering them so helpfully and in such a detailed way. And yeah, I hope everybody who joined us today has a lovely evening, and we'll finish by saying bye-bye.
Thank you. Bye.